Thank you all so much for coming today. I'm really excited to be here. I've had some really fun meetings already this morning, and I'm looking forward to getting to meet the rest of you. So just so I can get a little bit of a sense of the group of folks that I'm talking to, can I get like a little show of hands for people who identify or affiliate with the Complex Adaptive Systems School? Okay, cool. And then what about folks involved maybe in global futures or ocean futures, or consider yourself a biogeochemist? Awesome. Okay, so that's kind of exciting news for me because I see a fair group of biogeochemistry and a fair group of complex systems, but I didn't see quite so many who are both. So that means that I'm doing my job by bringing y'all together. And so hopefully after this talk is over, you can all make friends and start to collaborate. And for the complex systems folks in the room, I just want to reiterate the importance of the ocean and the coupled ocean climate system as an excellent case study for applying all of the principles that we use, because not only historically have a lot of the major innovations in theory and concepts that we use in the analysis of complex systems now come from the oceans, all the way going back to Volterra and generalized ecological network theory from managing fisheries through to the Lorentz system, to even most recently, the innovations in stochastic resonance and multifractal turbulent fluid flow that led to the 2021 Nobel Prize for Parisi. But also being able to understand and predict how the ocean is going to respond to anthropogenic perturbations, both to biogeochemical cycles and also to biodiversity and ecosystem management, is going to be crucially important to having a sustainable future and managing our changing climate. So in particular, I'm interested in our cohort of marine microbes that facilitate major transformations of carbon around the planet, and particularly from the ocean surface to the deep ocean where it can remain for a long time. So this schematic here is from one of the older IPCC reports, and the main thing that I want to show you here is that this little marine biota box here facilitates this transition via organic carbon from the surface ocean stock that interfaces with the atmosphere to the deep sea, which is our largest planetary carbon reservoir that's not in the deep subsurface of the planet. And in fact, even though the actual standing stock of marine biomass is very small, this pump that they facilitate is what is contributing to the stable accumulation of carbon in the deep ocean that exceeds, as of this reporting at least, 10 times our known fossil fuel reserves. So understanding how this flux works and what modulates its efficiency and magnitude over time is going to be critical to understanding what the fate of all of the carbon emissions that we've made historically and are making now is going to be. But this schematic, of course, is overly simple. And in fact, the real ocean is extremely heterogeneous. And so this is data collected from satellites as of late last night that shows the global distribution of chlorophyll across the ocean surface. And so you can sort of think of this as a way of distinguishing very biologically productive areas that are green on this map from less biologically productive areas that are the blue areas. And so you can see there's all sorts of interesting spatial structure in the distribution of organisms across the ocean that arises as the interaction between the complex geophysical fluid flow that determines the ocean circulation, as well as how ecosystems interface with the energy and nutrients that are being supplied via that flow. And the other thing that I want to point out is even in these blue regions where it seems like they might be less biologically active, that doesn't mean that they don't also have a large cohort of diverse, various, and really compelling organisms that are fueling those ecosystems. So these are a couple of flow cytometry imaging photographs that I took at a cruise a couple of years ago, about 24-hour sail north of the island of Oahu. This is the camera that I was using. And you can see that even in this relatively blue area, there are still all sorts of diverse organisms here that embody all kinds of ecological interactions, from spatially mediated aggregating types of interactions through symbioses. And so understanding how those things scale up to be our global spatial biogeochemical pattern formation is the main goal of my research. And we can zoom in even further and look at all of the life that is even subvisual to this camera. So to give you a little bit of a frame of reference, this is an epifluorescence micrograph of a drop of seawater. This really long red line here roughly corresponds to like one of these diatom cells in this image to give you a sense of scale. And all of these really bright green dots here are bacterial cells that have been stained with DNA dye. And then behind them, this entire field of all of these super tiny little dots that are everywhere across the entire view of the image are viruses. And so in a milliliter of seawater, there can be anywhere on the order from 100,000 to 1 million viral particles. And so it's the interactions between these larger eukaryotic organisms, bacteria and archaea, but then also all of these myriad viruses that actually drive the big scale ecosystem functioning. And how we get there is through all kinds of metabolic interdependencies and metabolic networks. So for the folks who have had to brave KEGG before, this might be a familiar image. But the reason I'm throwing this up here is just to show I've colored different parts of this metabolic network here by different organisms that do those reactions in a marine ecosystem that I've studied. And so you can see in order to like fill out this metabolic map, it actually requires a whole lot of interdependencies and interactions between diverse organisms. And only through the big scale functioning of that entire metabolic network do we get the emergent properties like the biological carbon pump that I was mentioning earlier. These metabolic networks, of course, don't operate in a vacuum. They operate in an environmental context that is forced by all sorts of complex physics. And whether it's on the sort of temporally local timescale of the daily cycle of light energy that goes into the system to fuel primary production, or at slightly broader timescales, seasonal complexity of the circulation that can change things like the degree of water column mixing, the temperature, or even the day length of the solar forcing. These kinds of like physical interactions across timescales are modulating the ecosystem functioning. And so that's what I'm going to talk a little bit about today, is how these different types of rhythms propagate through community metabolic networks to result in patterns of ecosystem level functioning. Yeah, so this is what we're going to try and figure out today. And I'm going to give you a couple of little vignettes of some different stories that I've worked on in trying to understand both bottom-up influences of how communities organize around nutrient uptake and competition, as well as top-down of how predators, or in this case viruses, coordinate with the community metabolic network. And so for our first talk today, talking about nitrogen uptake in the nitrogen-limited North Pacific. This is work that was sponsored by the Simons Foundation Collaboration on Ocean Processes and Ecology. This is like a view of some, but not even all of us. It was an enormous team effort, but I wanted to give an extra special shout out to my two co-first authors on this work, Angie Boyson at Pacific Lutheran, and then Matt Harkey, who's at the Gloucester Marine Genomics Institute. And what we did here was in 2015, we went out and conducted sampling using what we call Lagrangian sampling framework in this region, a little bit north of Hawaii, next to the historical Hawaii Ocean Time Series station Aloha. And so how the Lagrangian sampling mode works is if you want to take an ecosystem time series in a fluid that is moving, you have to follow the fluid. And so what we do is on the onset of sampling, we deploy a buoy over the side of the ship like this, and then tell the captain to follow the buoy. And so that's how we follow a patch of water as it advects, and gives us this particular sampling track that we took. And in the case of this cruise, every 4 hours around the clock, so without sleeping, we took samples for community gene transcription for large phytoplankton, as well as for bacteria. And then working with some metabolomics folks. We also did a targeted quantified mass spectral analysis of about 100 different key metabolites, and working with some lipidomics folks, we did the same thing for lipids, and then quantified total ecosystem carbon and a couple of other macromolecular classes. And altogether, this sampling effort gave us a data set of over 65,000 different time series of either gene transcript from a specific type of organism or the concentration of a particular biomolecule. And so I wanted to know, how does the day-night cycle influence all of these 65,000 different things? And so the first thing that I did was just take a screen of which of all of these different gene transcripts or molecules had a 24-hour cycle in their abundances. And I did that with a non-parametric method because I didn't want to only think about 24-hour cycles that were necessarily symmetrical or had any particular functional form. And so what I come out with the other side is something that looks kind of like this. This is a little sampling of some of the time series that we found that were distributed not only across photosynthetic organisms, but also gene transcripts of heterotrophic bacteria and the abundances of membrane lipids, storage lipids, pigments, all kinds of primary and secondary metabolites. There's even a diel cycle in the small molecule that gives vanilla its scent that we detected in this data set. So it turns out that the 24-hour cycle propagates throughout all sorts of different modes of ecosystem functioning here. After spending like a lot of hours with my colleagues poring over spreadsheets of the lists of all of these things, we realized that we needed a better way to think about the structure and governing principles of what was causing all of those 24-hour cycles. So I implemented a method that's called self-organizing maps for grouping all of the time series together based off of similarities in what their shape was over our sampling time. I'll also note the University of Vermont Complexity Center just had a preprint that came out not too long ago that was extolling the virtues of using self-organizing maps for multivariate time series analysis, and I felt very hip that I had been doing it several years before that preprint came out. But the really exciting thing that we were able to learn using this method was that all of these different time series relatively neatly clustered into 4 different groups. And when we examined the archetypes, or if you're familiar with how self-organizing maps work, the codebook vectors of those 4 clusters, we saw that they basically corresponded to things that peak at dawn, things that are high during the daytime, things that peak at dusk, and then things that are high during the nighttime. And so that was the framework that we were able to use to start organizing all of these different myriad functions and different types of small molecules into groups that might have something to do with each other. And in particular, we can build up to the macro scale observed property of total ecosystem carbon content. So this was our direct measurements of community organic carbon. So this is a proxy for the total biomass in the ecosystem. You can see that it has a peak every day at dusk. And when we looked into our mass spectral data, what we saw is all of our metabolites and storage lipids that had a 24-hour cycle to them had a coincident peak at dusk as well, which suggests that we're able to identify the molecules that are contributing to this overall signature in community biomass. And then looking at an even finer level of detail, when we looked at the gene transcripts, we were able to pull out the daily cycle of carbon fixation and respiration, where this pink line that we have here in the middle are all of the genes related to photosynthesis that get turned on during the day, of course, when all of this organic carbon and these photosynthetic carbon storage products are increasing. And then at dusk, when all of those molecules start decreasing, is when we see an upregulation of transcripts having to do with central carbon metabolism and the respiration and breakdown of all of those different products. And then another kicker that we got out of doing this analysis was we found a consistent day-night cycle in sugar uptake transporters of heterotrophic bacteria that all peaked overnight, which is when all of these secondary products are being excreted by the photosynthesizers during the day. So that's sort of a good sanity check that our data makes sense and the methods we're using are working, that we're able to pull out the day-night cycle of photosynthesis. And so that gave us the courage to start to look at what other kinds of aspects of community metabolism also follow a day-night cycle. And in particular, instead of looking at day-night cycles where everything is doing the same activities at the same time, I wanted to know what metabolic functions had day-night cycles where different organisms were doing the same thing at different times from each other. So I constructed this analysis that took all of our different functional genes and asked how many different species or types of plankton had a day-night cycle in the expression of this particular gene. And then from there, I took all of the pairwise combinations of the times of day that they peaked from each other and averaged them. So the idea is like a 0 on this axis means that every single organism that had a 24-hour cycle in this gene was turning it on at the exact same time of day. The result of this analysis, when I scanned over all of the functional genes that we had in our data set, is something that looks like this. And so first of all, to orient you with what's going on here is we recapitulated the result that photosynthesis is all happening during the daytime, which are these photosystem-related genes, are these orange triangles here. And those have a low difference in peak time among the organisms, but have diel transcription in a lot of different organisms. So these are all of our different types of phytoplankton that turn photosynthesis genes on during the day. But we noticed that a lot of our genes that were mostly turned on at very different times of day, and so were not synchronized across the community, had to do with the uptake and then assimilation of nitrogen into amino acids in the cell. And because the ecosystem that we were studying here is extremely low nitrogen ecosystem, we think that actually this has to do with mitigating competition, right? So in the sort of altruistic spirit of we'll all take turns taking up the limiting resource instead of all having to compete with each other for taking it up at the same time. We can translate that into characteristic patterns of different types of functional groups. So the main result here was looking at nitrogen uptake and assimilation, as well as the relative abundance of ribosomal gene transcripts, so actually using the nitrogen to make proteins. There was a consistent sort of dusk and overnight peak in all of those gene transcripts for our photosynthesizing organisms, while for our heterotrophs, they were peaking in the morning instead. And so it's this sort of like two shifts in the utilization of resources. This harkens back to an old result from theoretical ecology for terrestrial systems called temporal niche partitioning that suggests that you can alleviate yourself from competition by using resources or growing at a different time from your competitors. And so we were able to show that it apparently happens over the day-night cycle in the ocean. So I'm going to take a quick little break for a sip of water. And if anyone has some questions about that specific work before I move on, I'd be happy to take one now. Okay. Otherwise, I can go ahead and move on to some of my Bermuda stuff, where I wanted to ask, how general is this mechanism? And is it something that's actually sort of theoretically underlying community metabolic function? So for this work, this is part of a big collaboration primarily with Steve Wilhelm's lab at the University of Tennessee, Knoxville, but then also in no small part facilitated by field work we conducted out at the Bermuda Institute for Ocean Science on the research vessel Atlantic Explorer. So a big thank you to BIOS and the crew of the AE for facilitating that field work. What we did here was ask basically the exact same question, except in a system that had a different type of nutrient limitation. So instead of the North Pacific by Hawaii, where we were just studying, that's primarily nitrogen limited and has relatively higher phosphorus concentrations. Instead, we were in the Sargasso Sea, which is this super dark part here that has extremely low phosphorus concentrations and is considered to be persistently phosphorus limited. We did basically the same kind of analysis of the Lagrangian field track, throwing the buoy over, and sampled over a slightly longer time series this time. You can see these are readings from when we were collecting our samples. This is basically a proxy for the particulate carbon, the beam attenuation, and then the chlorophyll fluorescence. And so you can see the nice 24-hour cycles in both of these that, again, reflect photosynthesis during the day and respiration at night. For this one, we sampled more or less the same things, but in addition to the gene expression of cellular organisms, we also did a bunch of virus-selected metagenomes so that we could look at the diversity of viruses and pair those with our RNA sequencing. So we could not only look at what viruses were there, but what times their genes were being expressed, which is an indicator of an active infection. And altogether, instead of the 60,000 time series we had last time, we ended up with like over 100,000 different ones this time. Applying the exact same set of methodologies and doing the same corresponding analysis of how synchronized or desynchronized are particular functions across the community. Again, we get the same result that all of the photosynthesis stuff and genes that are related to cell cycle are all expressed on a 24-hour cycle by a large number of taxa, and they're all expressed at relatively the same time, which is a good consistent finding. But then we noticed that our nitrogen assimilation genes that were the really partitioned ones in our last study were actually slightly less so. And some of these, even these are components of the GS-GOGAT system here that I've labeled, were actually synchronized as opposed to desynchronized, which suggests that the nitrogen time of day partitioning isn't like a fundamental physiological thing. It's an emergent trait of the community dynamics. And instead, when we looked at our really desynchronized genes, what we see are a bunch of transporters that are involved in the uptake of different kinds of phosphorus species. And so we get this cool kind of hierarchy where even though nitrogen concentrations are also low in the Sargasso Sea, they're relatively higher than the phosphate concentrations. And so there's sort of this like direct sunlight, completely no partitioning, uptake of a not necessarily limiting nutrient, not very partitioned, and then uptake of a limiting nutrient is very partitioned. So this gives us some confidence to think that this mechanism of temporal niche partitioning is actually something that's more general, facilitating coexistence and alleviation of competition in these open ocean microbial ecosystems. And then furthermore, looking at the phosphorus transcript dynamics, we can see the same kind of separation of functional groups that we did for the nitrogen uptake in the last study, where sort of at dusk is when our cyanobacteria are taking up phosphate. We have some larger eukaryotic organisms that are taking it up during the day. And then all of our heterotrophic organisms or our mixotrophic dinoflagellates are taking them up in the morning. So that's my bottom-up spiel, is to say that the day-night cycle actually mitigates all sorts of different kinds of community metabolic activities and not just directly photosynthetic ones. And that the day-night cycle is also something that arranges coordination to alleviate competition for limiting resources. Now I'm going to talk. Yeah, go ahead.
So if you would, we have a project, I don't know whether Mike told you about it, we investigate the consequences of potential bioengineering,
yes,
geoengineering questions. If you would sort of increase the phosphor, the nitrogen in the respective domains,
yes,
would you get higher peaks?
So I think that's an excellent question and would make for a very good experiment that we could absolutely do. The other thing that I'll mention about this specifically for the geoengineering context is I was telling Michael earlier, phosphorus-limited ecosystems, in addition to taking up inorganic phosphorus, will also start to use organic phosphorus-containing molecules called phosphonates. And in hydrolyzing those to get the phosphorus out, they actually produce methane. And so if you end up, exactly, yeah, so you might end up in a regime where you accidentally swap out your CO2 for methane and end up in a worse scenario than you thought you would otherwise. Yeah, that's an excellent question. Yeah. And I can go ahead and talk a little bit about viruses. This is very exciting. This is kind of newer results. And so the first thing that I'm going to talk about is just mentioning also part of this field endeavor. Matt Sullivan's lab at Ohio State University was in charge of the virus metagenomes that we took our transcriptomes and mapped to get sort of the custom local suite of viruses. And so when we did that, it turns out that actually the viruses have a very strong day-night cycle in them as well, in terms of their relative gene expression. And since these are DNA viruses, in order to have RNA transcripts of a viral gene, that means they have to be infecting a host because they can't transcribe genes by themselves without a host. And so what we saw is actually like a very large number of bacteriophage transcripts that had a day-night cycle. You can see is like comparable to even the cyanobacteria that are doing the photosynthesis in the first place. But interestingly, they're all peaking at nighttime. So this clock is sort of our 24-hour clock of different peak times. And you can see this huge bar here is all at midnight, which suggests that like all of our 24-hour virus gene transcripts have the same peak timing as each other. Meanwhile, I also looked at gene expression for viruses of eukaryotic organisms. So those are bacteriophages that infect bacteria, but are giant viruses of eukaryotic organisms. There also were a fair number of genes that had a day-night cycle, except they were sort of spread throughout different times of day. They weren't quite as synchronized with each other. So I wanted to know, is there some sort of fundamental difference in the strategy between the bacteriophages that appear to have like a very temporally localized and very synchronized infection pattern, or the viruses of the eukaryotes that appear to have a different distribution of genes with day-night cycles? So I took all of those eukaryotic virus genes, and it turns out that they belonged to basically 3 different genomes. And these 3 genomes that belong to the Mimiviridae family, which if you're familiar with giant viruses, Mimi virus was one of the first big ones that was discovered of amoeba from some septic water tank in Central Europe somewhere. So these are relatives of that one. And I looked at the distribution of all of their different genes over the course of the day-night cycle. It turns out that none of them had any genes that peaked in the morning. So that seems to suggest that those viruses are not doing active infections in the morning. But then when we look at the whole picture, I could go through the molecular details with you if you're interested some other time, but basically what I'll tell you is when we group these genes into genes that are associated with the first part of infection, the middle part of the infection, and the end part of the infection, we recapitulate a cascade where these cool color genes, the green and the blue ones, are the early genes that turn on first. And then around dusk, there's a transition to some of the later genes that have to do with actually assembling the new viruses. And then at midnight is when we have expression of all of the genes that have to do with actually killing the cell and lysing it and getting out. So it turns out that even though like looking across all of the different genes, these viruses appeared to have sort of a different day-night cycle than the bacteriophages, it turns out the cycle is actually the same. It just takes them a little bit longer to get it going. So that suggests that midnight is sort of this really scary time to be a phytoplankton because it's when all of the viruses are on the attack. And it turns out that that probably has some biogeochemical significance. So there's this mechanism that's called the viral shunt, where when a cell has been infected and is lysed, there's the progeny viruses that were created from the infection, but then everything that's left over in the cell just gets reinserted as dissolved organic matter into the water. And that dissolved organic matter oftentimes also can be very rich in other kinds of nutrients like nitrogen and phosphorus. And so when it gets remobilized because it's no longer on the interior of a cell, heterotrophs can break it down. That provides more inorganic nutrients that can then stimulate the next generation of primary producers. So it creates this really tight recycling loop of essential nutrients in this kind of environment. So having all of the viral lysis events being localized in time probably magnifies the effect of this viral shunt mechanism. So you get like one big pulse of DOM at midnight when all of the cells are lysed, as opposed to a little dribble over the course of the whole day. So now I'm going to take like a little bit more of a macro view of that and look at how seasonal cycles influence this viral recycling. And so this is work from the same cruise, but then I also wanted to give a special shout out to Debbie Lindell's lab at the Technion in Haifa. And in particular, her student Camelia, who in this photo I found is looking incredibly dubious off to the side. Who did a lot of our work with the molecular quantification of viral particles for this particular story. So when we were out doing our cruise, we weren't just taking water from the ocean surface, we were doing profiles over the entire water column. And we found this really cool structure that emerged that right below the layer that's constantly being stirred and exposed to the surface by wind-driven mixing, which is marked by the white lines on these contour plots here. There was this one particular layer that wasn't very far, or wasn't very wide. It's maybe only about 5 or 10 meters deep. That is significantly enriched in oxygen compared to both the overlaying and the underlying waters. And it turns out that it's also associated with a big peak in the total amount of particulates, which suggests that there's a lot of biological activity that's going on in this particular layer. And so to give you a sense of what that would look like for sort of one discrete slice of all of these contour plots, we have the surface where everything is super homogeneous because it's all being mixed by the wind. And then directly underneath it, there's a big bump in particulates, but then also in the total amount of oxygen as the temperature starts to decline and you get into like a new density layer. And then there's some more aspects of the water column structure that are directly underneath, or that are a little bit deeper underneath that I won't get into today. Yeah, so the first question that I wanted to ask is, was that something that was just particular to the one cruise that we happened to be there, or is that like a structural feature of the Atlantic Ocean? And so using the Bermuda Atlantic time series run by BIOS, I went through their climatology of 30 years worth of some like 7,000 CTD casts looking at oxygen profiles over the course of the year. And it turns out that in fact, this is a recurring phenomenon where in the late summer and sort of early fall months, when the water column re-stratifies after being deeply mixed in the winter, there's this pocket of oxygen supersaturation that emerges sort of directly beneath that mixing layer. And that's something that's consistently been there for the past 30 years. So we wanted to know, what is the ecosystem like there? Is it different from what's going on in the surface or not? And so these are little profiles of cell counts from our different layers that we looked at. And it turns out in this oxygen maximum layer, there's a huge increase in the total number of cyanobacteria. This Prochlorococcus is sort of the grass of this ecosystem, as well as in heterotrophic bacteria. While for other kinds of primary producers, they don't have that same enrichment in that layer. So Prochlorococcus is extra happy in that layer for some reason. Then we also quantified the number of viruses that are infecting Prochlorococcus. And it turns out that there's an enormous increase in these T7-like cyanophages, the specific type of virus that we're interested in investigating here in this subsurface oxygen maximum. So not only is there a huge increase in the stock of potential hosts for these viruses, the Prochlorococcus, but then there's a huge increase in the actual number of viruses as well. And when we looked at our gene transcription data, we saw, in addition to an increase in the total number of those viruses, we saw a really big increase in the number of gene transcripts, which suggests that there are a lot of infections that are happening. And then for the heterotrophic bacteria that also had a huge increase in their numbers in this layer, we saw upregulation for organic nitrogen substrate transporters. So things that would come out of a lysed cell being taken up into the cell and then remineralized. And then for the Prochlorococcus, the primary producer, we also saw a big increase in inorganic nutrient transporters as well. So that suggests that this layer where oxygen is accumulating might actually be being driven by this viral recycling, where you get basically enhanced levels of nutrient retention and recycling that can facilitate more primary production, leading to more carbon fixation and more oxygen accumulation. And so that might be what's causing this sort of like decadally stable annual feature in the water column. And so I went back to historical data from bats and tried to look at the seasonality of the relationship between the Prochlorococcus and the total number of viruses. And it turns out that if you look in the months where this layer is not in the water column, there's not a significant relationship between the number of hosts and the number of viruses. But when the water column is stratified, suddenly the scaling relationship coalesces, and it turns out that the samples that have both higher numbers of Prochlorococcus as well as higher numbers of viruses are also the samples that have the most oxygen in them. So it turns out that, like, looking retrospectively, it turns this mechanism of sort of enhanced primary production through viral recycling is probably supported. And just to remind you, if we look at like the entire climatology of oxygen data, these same months where there's a significant relationship between Prochlorococcus and viruses, the sort of August through November period, is also the peak in subsurface oxygen saturation across the entire climatology. So that suggests that this oxygen maximum actually represents some sort of phase transition in the biogeochemical functioning of that ecosystem that's mediated by viral infection and recycling. And so just as like a quick little wrap up to talk about the stories that I shared with y'all today. So when it comes to nutrient uptake, taking turns over the course of the day is the strategy. And depending on whatever the limiting nutrient is in your local environment, that's how you decide what you're taking turns on. And then viruses, it turns out, all have a synchronized infection cycle with each other, where midnight is a viral infection hotspot for these ecosystems, and that can have really important biogeochemical consequences, such as the seasonal emergence of this high primary production, tight recycling, oxygen accumulating layer. So I'll leave you there, and then just if you want to catch up with me later, shoot me an email. These are some other topics that I also do work on that I didn't want to get into for this specific talk today. But that includes, I've recently become interested in exobiology and modeling non-Earth biogeochemistries that I've been working with Sarah on. And I have a bunch of other work on host-virus interactions and some stuff about how scaling theory interacts with all of these big ecosystem scale processes, particularly how they relate to ocean carbon uptake.